Welcome to the Veterinary Physiology online class. Today, we will be diving into Unit 2, which covers the digestive and respiratory systems. Our speaker for this session is Dr. Anamika Pandey from gnpsirclasses. Depending on their food habits, animals can be classified into different categories such as carnivores, herbivores, and various types of fermenting animals. For instance, ruminants like cattle and sheep have a complex stomach system, while non-ruminants like horses have a simpler digestive system. The simple stomach is divided into three regions: cardiac, fundic, and pyloric. Each region has specific functions, such as the fundic region containing gastric glands that secrete essential digestive substances. Gastric motility is primarily controlled by reflexes and neuroendocrine mechanisms. The vagal nerve and CCK play significant roles in this process, ensuring the proper movement of food through the digestive tract. Vomition, or emesis, varies among different animals. Carnivores and omnivores, except rodents, vomit easily, while ruminants rarely do. The neural center for vomition is located in the medulla. The motility of the small and large intestines is controlled by electric slow waves and neuroendocrine mechanisms. Key hormones like gastrin and CCK stimulate intestinal motility, while secretin inhibits it. Different animals have varying gastrointestinal structures. For example, dogs have a simple stomach with a small cecum, while pigs have a sacculated stomach and cecum. Sheep and goats have a sacculated rectum, which results in pelletized feces. The gastrointestinal function is regulated by neural mechanisms, including the autonomic and enteric nervous systems. Vagal and sacral nerves are crucial for this control, with acetylcholine being a major neurotransmitter. Various hormones play essential roles in gastrointestinal function. For instance, gastrin is secreted from the stomach and duodenum, while secretin and CCK are secreted from the small intestine, each having specific functions in digestion. Salivary secretion involves three types of glands: parotid, submaxillary, and sublingual. These glands produce saliva, which is crucial for digestion. The secretion is controlled by both sympathetic and parasympathetic stimulation. Gastric acid secretion occurs in three phases: cephalic, gastric, and intestinal. Each phase is stimulated by different factors, such as sight and taste of food, and plays a vital role in the digestive process. Pancreatic secretion involves the release of zymogens and enzymes essential for digestion. This process is regulated by neuroendocrine mechanisms, ensuring the proper breakdown of carbohydrates, proteins, and fats. Biliary secretion provides bile acids necessary for fat digestion. These acids are synthesized in the liver and stored in the gallbladder, playing a crucial role in breaking down dietary fats. In monogastric animals, digestion and absorption involve various enzymes from the salivary glands, gastric mucosa, and pancreas. These enzymes break down food into absorbable nutrients. Carbohydrate digestion starts in the mouth and continues in the small intestine. Enzymes like amylase and sucrase break down carbohydrates into simple sugars, which are then absorbed into the bloodstream. Protein digestion begins in the stomach with the action of pepsin and continues in the small intestine with the help of pancreatic enzymes. These enzymes break down proteins into amino acids for absorption. Fat digestion requires both pancreatic and biliary secretions. Bile acids act as detergents, breaking down fats into smaller molecules that can be absorbed by the intestinal lining. Ruminants have a unique digestive system with a non-secretory forestomach and a secretory stomach. The reticulum, rumen, and omasum serve as fermentation vats, aiding in the breakdown of complex plant materials. The rumen is a fermentation chamber containing billions of microbes that help break down food. This process produces volatile fatty acids, which are a primary energy source for ruminants. The rumen maintains a specific pH and temperature to support microbial fermentation. This environment allows for the breakdown of complex carbohydrates and proteins, providing essential nutrients for the animal. In the process of digestion and absorption, non-ruminants and ruminants handle carbohydrates differently. Non-ruminants use digestive enzymes to break down carbohydrates into glucose in the small intestine, which is then absorbed into the bloodstream. Ruminants, on the other hand, rely on microbial fermentation in the rumen to convert carbohydrates into volatile fatty acids, which are then absorbed into the bloodstream. Cellulose fermentation involves primary cellulolytic bacteria that act on cellulose, hemicellulose, fructoxans, and pectins. Methanogenic bacteria also play a role by producing methane, Colorado-2, and volatile fatty acids. The fermentation of starch is performed by primary amylolytic bacteria, which have a faster fermentation rate than cellulolytic bacteria. Volatile fatty acids, such as acetic, propionic, and butyric acids, are produced during fermentation. These acids are absorbed through passive and facilitated diffusion. Propionic acid is unique as it is used for gluconeogenesis. Acetate is taken up by tissues to form acetyl county A, which is used in citric acid cycle. Non-ionized volatile fatty acids diffuse more readily across cell membranes than ionized forms. VFA absorption mainly occurs through simple diffusion of the non-ionized form across the rumen epithelium. The concentration of non-ionized VFA in the rumen contents is low, favoring absorption into the bloodstream. Rumenoreticular motility involves biphasic and monophasic contractions in the reticulum and ruminal sac. The primary cycle includes both types of contractions, while the secondary cycle, or erectation cycle, occurs at the end of the alternate primary cycle. Normal rumen contraction rate is 1-2 per minute. Ruminants spend around 10 hours a day ruminating, with the highest incidence occurring in the afternoon and middle of the night. A diet low in roughages or finely ground roughages reduces rumination time. Avian digestion has anatomical differences such as the presence of a crop in the esophagus, a muscular stomach for grinding, and a glandular stomach for secretion. Birds also have a paired scum for microbial digestion and a bilobed gallbladder. The respiratory system consists of an inspiratory phase followed by an expiratory phase. The diaphragm separates the thorax and abdomen, requiring greater effort for inspiration. Expiration can become passive. Respiratory patterns vary among species, with horses having a unique biphasic breathing pattern. Breathing terminology includes terms like eupnea for normal breathing, dyspnea for difficult breathing, and apnea for cessation of breathing. Tachypnea refers to rapid breathing, while bradypnea indicates slow breathing. Alveolar ventilation is the rate at which new air reaches the alveoli. Dead space refers to the air that does not reach the gas exchange areas. Physiological dead space includes alveoli that are not functional or only partially functional due to poor blood flow. Tidal volume is the amount of air breathed in or out during quiet respiration. Inspiratory and expiratory reserve volumes are the additional air volumes that can be inhaled or exhaled beyond normal respiration. Total lung capacity is the sum of all lung volumes. Vital capacity is the maximum volume of air exhaled after maximal inhalation. Functional residual capacity is the air remaining in the lungs after normal exhalation. Closing capacity is the volume at which small airways begin to close. Gas diffusion is influenced by factors such as surface area, pressure differences, and tissue thickness. Surfactant, produced by type 2 alveolar cells, reduces surface tension within the alveoli. Surfactant deficiency in horses and swine can lead to respiratory issues. Lung compliance measures the distensibility of the lungs. Pressure changes during breathing include pleural, alveolar, and transpulmonary pressures. Surface tension within the alveoli is reduced by surfactant, aiding in lung expansion. Alveolar epithelium acts as a barrier against environmental insults and regulates water and ion transport. Type 1 alveolar cells cover most of the alveolar surface, while type 2 cells produce surfactant and can differentiate into type 1 cells for repair. Oxygen is transported in the blood, mostly bound to hemoglobin. The oxygen-hemoglobin dissociation curve shows the relationship between oxygen saturation and partial pressure. Hemoglobin's affinity for oxygen changes with different conditions. A small percentage of oxygen dissolves in plasma, while the majority binds to hemoglobin. At 100 mm of mercury, 97.5% of hemoglobin is saturated with oxygen. The total oxygen transported in the blood includes both dissolved and bound forms. Oxygen and carbon dioxide diffuse across the pulmonary alveoli and capillaries. The diffusion coefficients for these gases vary, with oxygen having the highest coefficient relative to other gases. The oxygen dissociation curve shifts to the right with increases in H+, PCO2, temperature, and 2,3 bpg. This shift indicates a lower affinity of hemoglobin for oxygen, facilitating oxygen release to tissues. Fetal hemoglobin has a higher affinity for oxygen than adult hemoglobin, allowing efficient oxygen transfer from mother to fetus. The fetal oxygen dissociation curve is shifted to the left compared to adults, indicating higher oxygen binding at lower pressures. Low pH, high temperature, and increased 2,3 dpg levels shift the oxygen hemoglobin dissociation curve to the right, decreasing hemoglobin's affinity for oxygen. Carbon monoxide binds to hemoglobin more strongly than oxygen, leading to poisoning. Colorado 2 and O2 bind to different sites on hemoglobin, and myoglobin in muscle cells binds oxygen more tightly than hemoglobin, aiding in oxygen storage. Carbon dioxide is transported in the blood in three forms: dissolved in plasma, chemically bound to hemoglobin, and as bicarbonate ions. The majority of CO2 is transported as bicarbonate ions, which play a crucial role in maintaining blood pH balance. Increased acidity in the blood causes hemoglobin to release oxygen more readily. This is because acidic hemoglobin has a lower affinity for oxygen, facilitating oxygen delivery to tissues. The Bohr effect describes how CO2 and hydrogen ions affect hemoglobin's oxygen binding affinity, while the Haldane effect explains how oxygen levels influence CO2 binding. These effects are crucial for efficient gas exchange in the lungs and tissues. The inspiratory center in the dorsal respiratory group controls the basic rhythm of breathing, while the expiratory center in the ventral respiratory group assists in forced expiration, especially during exercise. The pneumotaxic center in the upper pons regulates the transition between inhalation and exhalation, while the apneustic center in the lower pons promotes deep inspirations and regulates breathing patterns. Central chemoreceptors located on the ventral surface of the medulla are sensitive to changes in hydrogen ion concentration, influencing respiratory activity to maintain pH balance. Peripheral chemoreceptors in the carotid and aortic bodies respond to changes in blood oxygen, carbon dioxide, and pH levels, playing a key role in respiratory regulation. Periodic breathing involves cycles of deep and shallow breaths, with Cheney-Stokes breathing characterized by a gradual increase and decrease in breathing effort, often seen in certain medical conditions. The lungs have immunological functions, including acting as a barrier and producing antimicrobial substances. They contain various immune cells and structures that help protect against infections. Thank you for watching. Don't forget to like, comment, share, and subscribe to GNP Sir Classes official YouTube channel. See you in the next video.